we're pretty much ready to make a, yeah. So at this point, I actually need a large pan. Talia, could you hand me one of those cat pans? Because we need a firm surface. This brain knife, folks, is a specially designed piece of steel that is wedge-shaped. It's much thicker on its superior margin and very, very thin. And so it's designed so that as you begin your cut and then you drag, it just makes the tissue separate away as you make the cut, so you don't end up with lots of pieces, in little shards in between. So I'm going to turn this actually this way because I don't want the edge. And what we're going to do is I'm going to start posteriorly and then I'm going to drag, and I want to cut just lateral to mid-sagittal. Why? Because I want the midline still intact. If I cut right on the midline, my midline is destroyed. So I'm going to, on purpose, and what I have to really cut through is that corpora cavernosum. Okay, sorry. So I'm going to just stay a little bit lateral to that, and for the cerebellum as well. Sorry, let me make sure I don't. Okay, so that's kind of scissoring on me. Okay, so I'm going to stay just a little bit away from mid-sagittal if I can, and once in a while it rolls. So okay, and I'm going to drag so that I try to make as clean a cut as possible. And that dura is really, really tough. Okay, so I didn't get quite all the way through it, but I'm close. That dura is just going to hold it together a little bit. So, okay. So what I have, folks, now you can see the bright white corpus callosum, that really bright white structure. And I did get just lateral to midline because see what I can do with my probe? I can go under the corpus callosum on this side, and I'm actually in, oh, that's that probe that's so gimped. Sorry. I'm in the right lateral ventricle here. And I'm just, and as I get into that right lateral ventricle, here's what I can pull out of it, folks, and here's what makes cerebrospinal fluid. See this structure that I'm kind of pulling out of the space that's under the corpus callosum? I'm pulling out something called choroid plexus. What choroid plexus is, folks, is a highly coiled little tiny capillaries. And because they have such great surface area, what they do is they filter the blood. The fluid of blood, not the cells, not the red blood cells, white blood cells, and platelets, but the plasma of blood filtrates. You know what filtration is. It's movement of substances down their concentration gradient, but with a pressure gradient. The pressure is the blood pressure, and fluid seeps out. Now, are there some small proteins and glucose, and because that's cerebrospinal fluid is made as a filtration of the blood by choroid plexus in the ventricles, and the right lateral ventricle we can see into, can't we? And that fornix, that way deep recessed area of the where did I put the ventricular system? You can actually feel the probe go way back in there, and I'm not pushing the probe in. You can feel it go way back in there. Now, why can't I see into my left lateral ventricle? Because, folks, there's a membrane called the septum pellucidum. And if you did the reading, the septum pellucidum is right in the midline, and it acts as a membranous valve in between the right and the left ventricles. Because will your blood pressure always be the same on both sides? What if somebody has a little clot and the pressure, a little plaque, I should say, maybe not a clot, but a plaque, or they have narrower blood vessels on one side than the other, then they might make more cerebrospinal fluid on one side than the other. So this septum pellucidum goes back and forth to equalize the pressure. And that septum pellucidum is still intact here. I did make a little inroad into it, but you can see on this side clearly you're looking into the ventricle because all of the septum pellucidum is still on the left side. Now I can kind of rip into the septum pellucidum so that we could see into the left lateral ventricle, and we'd pull out choroid plexus on that side as well. There it is, it's just not so darkly stained. Okay, so now, once you've got your mid-sagittal section here, now we do the four major parts of the brain. We've got the big brain as the cerebrum. Do you see the little brain here? What was the white matter of the cerebellum called? Remember the arbor vitae, folks, the tree of life. It's called the arbor vitae. And the gray matter of the cord you can actually sort of see as little leaves called
folia.
Folia. Very good. It's the analogy of the tree of life. So okay. So then we said that the fourth ventricle, folks, the fourth ventricle is full of choroid plexus. Look at this. The fourth ventricle sits right at the base of the cerebellum. And was I lucky enough to get central canal of the cord? I actually was lucky enough to get that. Yeah, because what happened was I stayed I was a little too far, lateral on the right side. But so there's the central canal of the cord, which indeed the fourth ventricle is continuous with that. Okay, so then cerebrum, cerebellum. Now let's do, what's the next part of the brain right under the cerebrum? It's called the diencephalon, and folks, it consists of the thalamus, which is this big round gray area. Because what is the thalamus, folks? It's just a bunch of nuclei, collections of nerve cell bodies. So there's the thalamus. It's almost perfectly rounded. And then I didn't get all the way through the dura. Let me go down a little bit deeper, because if our pituitary gland is right there. Oh, it's really too hard to cut this with a knife. Let me go with the scissors, so we'll just go like this. So now you can see, even though the pituitary gland is not part of the brain, it does hang off the hypothalamus, is that area of gray matter just inferior to the thalamus. So here's the pituitary gland, folks. Here's the hypothalamus, is just this region under the thalamus. And together, the thalamus and the hypothalamus constitute the diencephalon, which is the second principal part of the brain. So again, for you folks, there's the pituitary, there's the hypothalamus, there's the thalamus, and we can see it on this other hemisection as well. We can see the thalamus and the hypothalamus. We've got a lot of the sort of the association tracks running through it, but the hypothalamus is sort of down here, and then there's the pituitary. But it actually shows up best on this left hemisection, thalamus and hypothalamus. Then, after the diencephalon, what parts of the brain are there? It's called the brain stem, folks. It's called the brain stem. I know it was hard for me to remember. Okay, and the three parts of the brain stem are the midbrain, pons, and medulla oblongata. So, okay, so right after the diencephalon, folks, it's hard to sort of see, but the midbrain is all this, it's got gray, it's got nuclei in it, but it's mostly going to be white matter. So there's the midbrain, because remember, it's the place where incoming and outgoing tracks are, passing each other. So there's the midbrain. The pons is this poochy part. On a diagram, this is how I remember, the pons is always the part that has this pooch in it. See on this lateral view, there's the pons. So the midbrain, pons, and then the medulla oblongata only goes, it's oblong, so it's long, but it only goes to what landmark? To the remember the foramen magnum of your occipital bone? Because the minute it passes through the foramen magnum, it's now called the spinal cord. Very good. So we have see, there's the thalamus, there's the hypothalamus. So right after the diencephalon, midbrain goes from about here to here. This pooch is has been flattened, but you might get a brain that has a really nice poochy pons. Okay, and then medulla oblongata. You can actually see the area of stricture right about there, where this becomes the cord. But there's the medulla oblongata. Okay, so then couple of other landmarks, and then you're going to be set free to do your own brain. So folks, I didn't get this on this right hemisection. See this bright white structure right there? Let me show everybody. If I put our brain back together, what was that bright white structure? Was this little optic nerve. that you can just barely see right there. So now on hemisection, you've got the optic nerve there, and on this side, it's so oblique because I got some of the actual nerve itself, but there's the cross section of optic nerve. And then if you lay a, let's see, which side did I get it on? Yeah, I got it best on. It's not stained quite as nicely as some of you will have a much nicer, a darker staining. Actually, it's over here just as well. But if from the optic nerve, if you do a straight line across the thalamus, you'll see what's called the pineal gland. And the pineal gland is this little guy right through there. It should be a lot darker. I'm kind of disappointed that we didn't get more of a pineal gland, but we'll get it on another one. So you need to know the optic nerve, and the pineal gland sits atop the thalamus, and it's this kind of little guy right there. Okay, now we can also see. Can you also see the superior and inferior colliculi for on a cross section? So sometimes you have to go back to your 3D, but there's a superior colliculus and there's the inferior colliculus. Okay, that we named before. And beyond that, folks, again, you just be sure you kind of see the arachnoid mater and so forth. So what I'm going to do is let you with